Okay, good morning, everyone. Thank you for joining us on our session, which features two investigators from Scripps. We have Andrew Ward, who is a professor at the Scripps Research Institute working on vaccines. And we have Case McNamara, who's the director of infectious disease at Caliber. And we're each going to have them present in turn. And then at the end, we'll have plenty of time for some questions.
Good morning, everyone. And thanks for joining us for this session. Today, I'm going to give you a sort of a broad overview of where we are in terms of next generation vaccines. And the good news is the next generation is really happening in front of us, really in unfolding. And we at Scripps are pushing a lot of these innovations. And I'm going to run through some of the work that we're doing in collaboration with investigators at Scripps and elsewhere, and really highlight some of the challenges and the thinking that goes into how we design and prepare vaccines for existing pathogens and emerging pathogens. Just start with a really, classic example of a vaccine. So here's polio vaccine. It's very simple and highly effective, right? So here's polio. These little virions, if you inactivate them with some fixatives like formaldehyde, you cross-link and perturb the surface such that it can't infect, but it presents all of the proteins of the polio virus that are necessary to generate a strong antibody response. And so this works very well. And here's a nice electron microscopy reconstruction showing where the antibodies, in particular, these yellow antibodies bind on the surface of poliovirus, and where they bind is they overlap with the receptor binding site. So these antibodies are listed by almost universally by everyone, and poliovirus can't get into the cell because of this. So this is a really nice example. Of course, that doesn't work for a lot of the new pathogens and other pathogens we're dealing with, but the same principles hold. And one of the reasons, this is another nice example of why viruses or how viruses can use immune evasion techniques or avoid immune responses, and that's respiratory syncytial virus. So if you look at the virion here, you can see it's studded by these red and blue spike proteins. These spike proteins are responsible for finding the cells and driving the fusion process. Now, if you look, it's a combination of both these, what's known as the pre-F or pre-fusion F protein and the post-fusion F protein. The post-fusion F protein is highly immunogenic and immunodominant, and the antibodies that are elicited to the post-fusion aren't effective at neutralizing the virus. You need to hit the pre-fusion target in order to make a neutralizing antibody response. So this is one of the reasons why you can't just present RSV in terms of a vaccine, because all the antibodies will be biased in the wrong direction. So this really sort of sets up the stage for structure-based vaccine design. And so RSV-F, as well as HIV envelope glycoprotein, which several investigators at Scripps are working on. And this involves solving structures iteratively and then adding mutations like disulfide bonds or hydrophobic amino acids or prolines or other tricks to really arrest and stabilize this pre-fusion conformation, which is the immunogenically more favorable form. So there's a lot of excitement that in RSVF, that's going to be enough in order to bias the immune response correctly. In HIV, as I'll touch on, just stabilizing the pre-fusion state of the spike protein probably isn't going to be enough. So just to give you a high-level view of how we think about and how we develop new vaccines, we spend a lot of time collating known data, known structures, and spend a lot of time in the computer designing proteins, engineering them, as I showed, to be in certain conformations that are very stable and that present the epitopes that we want antibodies to target. As has really been highlighted by the COVID vaccine development. There's a lot of different innovations in delivery modalities. So, there's the classic inactivated vaccines, similar to the polio vaccine, as I mentioned. There's vectored vaccines where you can take adenovector, a different type of virus, and deliver a protein. There's recombinant proteins, as well as most of the success, particularly in the RNA vaccines. And then we spend a lot of time assessing the different immune responses in animal models. And this is really important because sort of vaccinology went from empirical, where you sort of got a phenotype of yes, it worked, or no, it didn't, to now we use next generation sequencing to look at the entire landscape of antibodies that are elicited. We use a lot of structural biology to define the sites that are hit. And from this sort of enriched information, we can go back and we can redesign. And so all of these areas are areas that my lab and others at Scripps are really interested in accelerating this process, because the faster we can design, the faster we can test, and the faster we can analyze, then we can sort of close that loop quite quickly. And again, the RNA-based vaccines are allowing us to accelerate quite quickly. So for COVID-19 or any of the SARS-CoV-2 vaccines, the first wave of vaccines were really, it was known that the spike protein on the surface was like those other viruses I mentioned, the target of neutralizing antibodies. And basically every different modality that was available was undertaken in the vaccine development process. As I mentioned, all of these different ways of delivering the spike. And it's really a wonderful test in vaccinology that's being borne out in terms of what works best. And we know that, there's been success with some ad vaccines, inactivated vaccines, and then, obviously huge success in the RNA vaccines. So, these deliver all of the viral components. These deliver just the spike protein. And so there's, again, like I said, the future of vaccinology is really compressing this timeline. And you can see from what happened with COVID-19 that this can be done actually in 6 to 12 months in a quite reasonable time frame, as long as we understand what the antigenic target is and how to make the protein stable, like in the cases of those others. And so one of the innovations that came out of my lab in collaboration with Barney Graham and Jason McClellan was in 2016, we solved the first structure of a human coronavirus spike. And this is from a seasonal virus called HKU1. And having the structure in hand allowed us to do that structure-based design that I alluded to for HIV and RSVF. So we had a strong basis for the logic of programming this conformation. And what we ultimately did after screening lots of different mutations was to introduce these 2 proline mutations in the fusion machinery. And this was enough to arrest the conformation in the pre-fusion in antigenically desirable form. It also had a nice byproduct of when we tried to express the protein with the wild type sequence, we got almost no level of expression. We added the 2P mutations, and here's for both SARS and MERS. You can see a huge increase in the expression levels. And then importantly, when we look at the wild type by using our electron microscopy imaging, you can see these rod-shaped in red, and these are the post-fusion antigenically undesirable. When you add the 2P, it brings you all back to pre-fusion. So this allowed many of the companies that were developing vaccines to incorporate those 2P mutations, Moderna, Pfizer, BioNTech, Novavax, Johnson & Johnson. And there's lots of different data. Of course, the 2P mutations weren't the only things tested, but they were selected for in the case of Novavax because they got higher expression and more stability. And Johnson & Johnson had superior immune responses, and so on and so forth. So you can see how pre-existing knowledge and a lot of the work that was done on other viruses really played into the ability to rapidly generate what turned out to be remarkably effective vaccines against COVID-19. So since then, there's been a lot more unpacked in terms of particularly where the neutralizing antibodies bind, right? So most of you probably know now that the receptor binding domain here in purple is the primary target of antibodies. There's all sorts of different classes. Ian Wilson, a colleague of mine at Scripps, has solved a number of these structures really in real time as they're being isolated. And you can see there are basically 4 different ways in which you can target the receptor binding domain. Certain ones like class 1 and 2 are really potent because they block the union with ACE2, the receptor, but they're also quite susceptible to a lot of the variants that are emerging. The class 3 and 4 are more resistant to the variants. And then class 4 is a potential for cross-reactive with other SARS viruses. So a lot of the work being done now is to make vaccines that are even smaller, that present just the receptor binding domain to immunofocus. Of course, the full spike may have other advantages, and I don't think that the full spike is going away, but this is just an example of how you take the knowledge and you zoom in and design the next round. And then even further, people are developing nanoparticle-based vaccines using these engineered proteins that form these self-assembling arrays. And the advantage here is if you just immunize with that receptor binding domain alone, you get weak B cell signaling. If you incorporate it into this nanoparticle with multiple copies, you get a much stronger B cell response. And so, again, here's where structure was used to design a lot of these materials that can be used to now present lots of different antigens in a plug-and-play format and get strong responses against viruses of interest. We're still looking, again, in real time for new antibodies. And here's just a new antibody structure from our lab showing that there are some antibodies that people generate that actually can bind and avoid the common escape mutations that are present in the variants. So it suggests that maybe you could even create an even more focused vaccine on this particular epitope and have the advantage that they escape variants. And of course, this antibody can be developed as a therapeutic. And Toscana Biosciences is doing just that because it's highly potent, it has favorable properties, and it avoids the mutations. So just to zoom out in a few closing slides, again, a lot of the logic and innovation that has gone into a lot of vaccine design is being driven by the work that is being done on HIV vaccine development here at Scripps. So in a consortium led by Dennis Burton, we're using lots of different structure-based design to elicit very specific and particular classes of antibodies. I mentioned that pre-fusion structure of HIV trimer. This is in humans right now. So it's the first sort of proof of concept for pre-fusion stabilized. And of course, it's not going to be the end of the vaccine development. But it's an important proof of concept. And then another colleague at Scripps, Bill Sheaf, has shown now that you can take, just a small piece of this HIV envelope protein and design it such that you elicit a very particular class of antibody down to the sequence level. So we have sequence level understanding of what people's antibody or immune repertoires look like. And then Bill and his team are designing antigens that elicit one particular class of antibodies, which is really remarkable. And they had quite a success recently in their clinical trial for the first time, showing that you could what they call germline target by designing specific entities and then immunizing. So this HIV remains, the toughest opponent we have, but it's forcing us to create new vaccine modalities, innovate, and then, of course, as I mentioned earlier, innovate in terms of the types of assays we use to measure the immune responses, next generation sequencing, high throughput structural biology. And so that's where, my lab, again, just to end on a few contributions, here's sort of the workflow, the classic workflow of serology, showing, if you immunize an animal, you can do some preliminary assays to test for binding and maybe efficacy. You start to generate monoclonal antibodies, and then the structural biology classically comes many weeks out or months out to years. We have a new assay now in the lab that really allows us to start the structural analysis very soon after the immunization to really delineate the total antibody response. And again, so that's an important way in which we can iterate quickly. All this is a lot of the structural work is done. If we were actually having the spectrum in person, we'd be on the Hazen campus, and that's where our wonderful cryo-EM facility is, with tens of millions of dollars of infrastructure and computation that allow us to do this amazing work in real time. That's really where our effort is being placed. And we're doing this not just for HIV, but we have programs in Ebola, malaria, influenza, coronaviruses as well. And so all of these principles can be used across the different viruses. And it's a really nice way to learn something in one field and bring it into another field. And just to end with a really pretty picture, this is basically all the antibodies that we've characterized against Ebola glycoprotein. This was my student Daniel Murin's thesis, and it really does show you the diversity in which antibodies bind, mechanisms of neutralization, and really principles for further design of maybe an Ebola-based vaccine. I mentioned my lab has been doing a lot of work characterizing what antibody responses to seasonal coronaviruses look like. We're now, of course, doing a lot of work to SARS-CoV-2 infection and in vaccination. And then we also solved the structure of the Novavax vaccine using cryo-EM. So cryo-EM has really allowed us to take images and pictures of things like vaccines and vaccine formulations like we haven't been able to before. So with that, I will end and acknowledge the great team of people in my lab that, of course, are doing all this work with very generous funding from the National Institute of Health, the Gates Foundation, and IAVI, and really a lot of internal support from Scripps Research. And you can see this is the last time we were able to get together to take a picture, because really this is how all the people in the lab are doing research now. So we do this with Zoom, as you know, but really the efforts have been quite remarkable from the scientists, not only in my lab, but across campus. And I'd like to just acknowledge, the hardships and otherwise that they've had to deal with to keep the train moving. And with that, I think I'll end and pass it over to Case.
So the focus of my talk is going to be a change of pace from what Andrew just presented. And I'll be laying out the assays that we put in place within Scripps Research to enable antiviral drug discovery against SARS-CoV-2. And so I wanted to start by giving a brief retrospective of where we were a year ago today, really, when the pandemic took hold. And the objectives in the field were to quickly identify new medicines that we could repurpose to quickly address the SARS-CoV-2 pandemic, which was growing. And so this is a very rational approach. And so many labs and institutes throughout the world took this approach. And hydroxychloroquine was one of the early repurposing candidates that created the most notoriety. And that was really reflected that by April 2020, you can see there was more than 200 clinical trials dedicated to looking at either hydroxychloroquine or chloroquine itself, involving 130,000 individuals. So really just a massive and incredible effort put around this candidate, right? We all know it ended up not being efficacious and isn't a good molecule for COVID-19. Conversely, if you look at remdesivir, which was a failed treatment for Ebola, this ended up being efficacious against SARS-CoV-2. And now is the lone FDA-approved molecule for a direct-acting antiviral. One of the limitations of remdesivir is that is IV infusion versus being orally administered. So ideally, we'd like to find something that would have more acceptance and the ability to administer more easily by having the oral drug. And that kind of brings me to the clinical pipeline. So early last year, there was a number of earlier stage compounds that were more custom designed towards SARS and even MERS that emerged as really good reposition candidates for SARS-CoV-2. Within those is EIDD-2801. This is being co-developed by Merck. And just earlier this week, there's some really promising phase 2A data that was released that indicated that this molecule is efficacious within patients. So bringing back around, what we did at Scripps Research, like the rest of the world, was do a very intense drug repurposing program. And this was funded by the Bill and Melinda Gates Foundation. And the reason one of the major reasons that we took this approach too is because that we have the Reframe library. Pete discussed this library earlier today. We consider it a best-in-class drug repurposing library. This library was funded by the Bill and Melinda Gates Foundation, in particular for neglected tropical disease, with the thought that we could identify very quick drug repurposing opportunities from that library, or even great tool compounds to help understand the biology of these other pathogens. So this is an open access library. And so we sent this library out to more than 30 external partners over the past year. And those data from those screens are actually posted on reframedb.org, and you can freely access that. What we did was internally set up a high content imaging assay in our BSL-3 facility. And this was enabled by a really wonderful collaboration with Tom Rogers here at Scripps and UCSD. And so what we did was take a wild type SARS-CoV-2 and infect HeLa cells that were expressing the ACE2 receptor. And again, all this work is done in very high biocontainment levels. So BSL-3 facility that is located in Scripps Research. So this really entails a lot of extra PPE and really limits the amount of people that can work on the project at one time. Despite those limitations, we very rapidly and quickly developed a high quality assay that enables us to screen thousands of compounds per week. And this was done, spearheaded by Melina Bukowski, who's a member of my team at Caliber, but also with incredible help from Tom and Nate as well. So our strength, particularly in my team at Caliber, is drug development, or I should say also assay development. And what Melina did very quickly was miniaturize this into 384 well format and convert this into a high content imaging assay. Through Tom, we were able to get a clinical isolate from the state of Washington of SARS-CoV-2, as well as gain access to convalescent patient serum. And that serum allowed us to actually visualize, to label and visualize the viral particles in the infected wells. So this assay itself runs for 24 hours, at the end of which we stain so we could now see the viral particles, as you can see shown here, as well as the host nuclei, so we could understand whether there's a cytotoxic effect of our compounds also against the HeLa cells. What's really great about this assay is that it's a very high quality, and the data turnaround time is very rapid. It's about 3 days or so from the time we start, from the time we get the images and run the algorithm to quantify the reduction, if any, in the viral particles, as well as the cytotoxicity. So what I show on the far left here is just a nice way to kind of drive home that we had a very high quality assay. We see great grouping of remdesivir and apilimod, which is a PIK5 inhibitor, as positive controls in the HeLa assay. DMSO was our negative control. That's the vehicle, so to speak, that all of our compounds are solubilized in. And pyramycin was just our positive control for cytotoxicity. It kills the HeLa cells, so it's highly active against everything. So screening Reframe, there was just a little under 12,000 compounds that we screened. This led to 61 hits. When we followed up these hits in dose response, we set a pretty loose criteria. And that was, we wanted EC50s less than 10 micromolar. That's a pretty low bar. We really wanted things that were nanomolar or even low nanomolar. When we further put the bar to include selectivity, so things that didn't really have any effect on the HeLa cells, so no cytotoxicity, that then reduced the number of hits of interest to 8. So just a handful of hits that we would consider potent and selective. The nice thing about the reframe library is that in addition to having access to compounds that we may not otherwise be available to us, is that we have data around these compounds that are either in clinical development or FDA approved. Sometimes we have pharmacokinetic data that informs on the exposure plasmas that we have. The route of administration is the oral, IV, infusion, topical, as well as the dose that's approved for or being used, the safety margins, and so on. So our ideal candidate would be very safe, orally administered, compound with a high plasma exposure, ensuring we get good exposure to the lungs and to help treat COVID-19. I'll say this, out of the 8 compounds that we looked at, none of them really ticked all these boxes and looked immediately as, just silver bullet repurposing agents. So what we did was expand this net, and we pivoted to include a second assay, and this was a Calu-3. So Calu-3s are derived from human lung epithelial cell line. And unlike the HeLa cells, they naturally express ACE2, along with TMPRSS2, which is a serine protease that cleaves the spike protein and allows a natural entry of the virus into the cell and release the RNA so it could lead to propagation or replication of the virus. Now, in the HeLa cells, there's more of a non-natural entry that occurs here. There is no TMPRSS to cleave the spike protein. So instead, it relies on cathepsins present in the endosomes to do that cleavage, and then ultimately you get release of the RNA and replication of the virus. So there's very specific inhibitors such as hydroxychloroquine that work only against viruses that go through this endosomal pathway of entry. And there's other inhibitors like nafanostat that only work against TMPRSS2. So these will be very specific to the assays. Now, the compounds that really target viral replication within the cell are going to be active in both assays. So something like remdesivir that targets the RNA-dependent RNA polymerase will show very comparable activity in each. So applying this Calu-3 assay to the reframe library, expectedly we got a differentiated hit list from that. So again, I'll point out halofantrin is one of the hits. This belongs to the 4-aminoquinolines. So this is from the same family as hydroxychloroquine. We see in the HeLa assay nanomolar activity. We see in the Calu-3, it's dead, it's inactive there. And that's not a surprise because it has a different mechanism of entry. Now, interestingly, if we look at some of these other inhibitors that we missed in the HeLa assay, essentially, now we see nanomolar or low micromolar activity for these hits within the Calu-3. So between the HeLa and the Calu-3 assay, we're able to generate about 10 to 15 compounds of interest to move forward in a cascade. So what I provided to you is some details around the screening assays that we put in place within Scripps research. What we did to really help validate these hits and really justify whether they should move into a proof of concept in vivo model is develop a primary cell model in which we can evaluate these compounds against human bronchial epithelial cell lines. So these are differentiated in the air-liquid interface. And so this assay is very time-intensive, resource-intensive. It takes 6 to 8 weeks to get to this point. But once we differentiate, they develop cilia, they start producing mucus. And these are cells that you're going to find, they're going to be more representative of the cells you're going to find in your lung, where we see primarily SARS-CoV-2 infection. And this assay has been quite powerful for us to help really validate and verify that compounds coming out either the HeLa or Cal-3 also show activity in this primary cell line as well. So that work has been led by Karen Wolf and Melanie Kirkpatrick, who are also part of our team at Caliber. In parallel, and very early on, Tom Rogers and Nate, and now John and Namir have been working very hard to develop and support the hamster efficacy model. So this model, interestingly enough, hamsters do develop COVID-19, and we can see this little chart here shown in panel B, that in the first 5 days, we see quite a bit of weight loss in these hamsters due to the SARS-CoV-2 infection. And more importantly is at the end of the assay, we could sack these hamsters, take out their lungs, process those lungs, and look at the viral load within the lungs. And for a compound here, this is EIDD 2801. This is one that just showed the favorable phase 2A output. We see it has an incredible reduction, almost no detectable virus particles at high doses. At a lower dose, it's intermediate, and in the vehicle, of course, there's no effect there. So we've been able to really leverage this in vivo model to profile the reframed compounds, as well as new compounds that we're developing in the medicinal chemistry program. What I'm pretty excited by too is some work done by John Taharo and Namir to develop a mouse model. Anyone who's worked with hamsters know they're chubby. They're much larger than mice. And so they consume a lot more compound. It's also a little bit easier to get PK from mice. So we're looking forward to including the mouse model to really supplement our drug discovery efforts. So unfortunately, even though we set this wonderful pipeline, the reframe compounds that we identified as potential repurposing opportunities have proven to be really moderate and not really compelling compounds for drug repurposing. So at this point, I think the take-home lesson that we've got from this effort is that we really need to custom design, custom tailor compounds towards SARS-CoV-2, and there isn't going to be quick wins from drug repurposing necessarily. But the beauty of having access to the reframe library and what it enabled us to do was to actually establish a robust and very collaborative drug discovery pipeline at Scripps. And so that library really represented a very rich collection of tool compounds and that allowed us to validate and set up the HeLa and Cal-3 screening assays, as well as a secondary assay in the HVAC model, and even move compounds into the in vivo model. As a result, what we've done internally is pivot to developing novel inhibitors targeting the viral protease and the RNA-dependent RNA polymerase as well. And we hope to have at least one preclinical candidate by the end of the year, and it'd be wonderful to be able to present on that in the future. Because of our high throughputness and really the awesomeness of the team working in BSL-3 to really execute on all these assays, we're able to support more than just our internal efforts. And we are, through the Gates Foundation, we've been supporting testing of numerous compounds from multiple big pharma partners as well. And we put those through the HeLa and even now the Cal-3, and a select set of those compounds will go into the HVAC primary cell model as well as the in vivo model if they're justified. So at this point, we all know that vaccines have astonishingly made it to market very quickly. And so you may be asking, why is there a need for a small molecule antiviral therapy at this point? And I think, as we're all aware, there are escape mutants that will continue to emerge that will threaten the efficacy of vaccines or reduce that. And there's just going to be a segment of population that will not accept the vaccines or don't have access to the vaccines. So for me, when it comes to neglected tropical diseases or any kind of infectious disease, we always want to have a war chest of tools available to us to help prevent and treat those diseases. So vaccines are at the top for prevention. And monoclonal antibodies are a wonderful tool as well for prevention and even treatment too. And I see a very strong role for oral small molecule inhibitors to also be used either in prophylaxis or even treatment. And we could even reformulate those inhibitors into long-acting injectables or even inhaled treatments to increase efficacy or to kind of meet different TPPs within that. So with that, I'll wrap up the presentation and just quickly go to acknowledgements. What I'm trying to drive home here is that this is a highly collaborative effort across the whole institute. Caliber, great virologists at Scripps as well that have all contributed to this. You know, at Caliber, we're very good at assay development and drug discovery, but we do need viral expertise through all the steps. And so Tom, Dennis, John, Juan Carlos, and others have all been there to help provide that to us, both on the in vitro and in vivo side. And of course, to do any kind of drug discovery, we have quite a large network and infrastructure at Caliber, both on the HTS, CNG, chemistry side, which is led by Arnav Chatterjee, and pharmacology, Sean Joseph and Ashley Woods, as well as Mitch Hull. So with that, I'll end the talk, and I think we could transition to the Q&A.
So we have one particular question, I think, that in a normal in-person event would be handled by discussions informally outside, but I think it's a great question. So we have Peter Heifetz, who's the CEO of Orpro Therapeutics, and he asked the question, is if you have some compounds that are potential antivirals, is there a way to collaborate to investigate their mechanism?
Yeah. Yes, there's always opportunities to collaborate and move forward. I think we're always interested. I think that's a conversation we can have separately offline, but the way Caliber works, and Pete alluded to this earlier, is we try to be highly collaborative. And as I alluded to earlier, I think we are very expert in drug discovery, maybe not always as expert in the biology inside, but within this, I think we try to keep a really wide net to bring as many things in as possible.
Okay, terrific. And I think one can always reach out to individual investigators and just initiate a conversation.
Absolutely.
I would stress that Scripps and Caliber are highly collaborative environments, and we welcome the opportunity to interact with scientists from industry. Maybe another question, Andrew. A member of the audience had a question about your class 1 through 4. And what does that really mean, class 1 through 4? And are those classes common to other virus spike proteins, or are these really specific elements to COVID-19?
Right. Yeah. So the classes just mean the location at which they bind. Right. So there are clusters of epitopes or surfaces on the protein. And in that case, I was showing the 4 major surfaces targeted by antibodies on RBD. They come from a lot of different genetic backgrounds, so a lot of different germ lines are being used, and some are more potent than others, and some have potential to cross-react, but the cross-reactivity is really with SARS and highly related viruses. So ongoing work by us and others is to find sites or epitopes, classes of antibodies as examples that are much more cross-reactive, and to develop a pan SARS type vaccine, a pan beta coronavirus vaccine. And this is where the structure and bioinformatics and all the work that's being done in sequencing is really driving our ability to chart out what is possible in terms of how broad a vaccine can be, and how should we go about developing that.
So, Andrew, I have a question for you about the role of glycosylation for COVID. So, in both flu and in HIV, the outside of the virus is heavily glycosylated, and this seems to be in part a way to combat antibodies from binding to the spike and allow it to actually enter cells. So, could you say something about, is the coronavirus glycosylated, and does that impact the efficacy of antibodies?
Great question, and it was actually a slide that I took out of the presentation in the interest of time. But so as you mentioned, HIV and flu are highly decorated with glycans, and in those examples, they are immune evasion mechanisms. So adding a glycan can knock out, a whole class of neutralizing antibodies, and that's very clear that they're doing that. For coronaviruses, even though they're highly decorated with glycans, and there may be some evolutionary history to immune evasion in the animal reservoirs that they've been present in humans, you don't see glycans, or it's very rare for glycan to be added as an escape mutant. And I think one of the reasons why is in order to engage ACE2, which is a large protein receptor, if you add a glycan, you then knock out binding to your receptor. So it makes it much more difficult for the virus to use glycans as an escape mechanism.
Right. Okay. Great. Now, so I have a question, maybe to engage the two of you. So, Case, you talked about developing antivirals, and Andrew focused primarily on vaccines. But there's another mode, which is antibody therapeutics, which can be used as a treatment. And the question I have, there are very different mechanisms of action. Is there any opportunity for synergy between a small molecule and an antiviral is there any opportunity for synergy between a small molecule antiviral and antibody therapeutic?
I'll let Case start.
Yeah, so you're saying if we co-administer the combination between a small molecule and an antibody, yeah, I think absolutely. I think where we focus more traditionally is to look at a combination of compounds with each other to hit multiple mechanisms of that viral pathway, to increase efficacy and to prevent resistance. And I guess we're looking at two different things. One, with a small molecule inhibiting that replication of the virus. But I think here, with the application of an antibody, you're also really neutralizing and controlling its ability on any successful particles to really reinvade. So I think together, there's of course the ability to put those together and pair those and put that forward.
Yeah, and I think, antibodies and small molecules have different biodistribution. So you might, get better results in the lung with an antibody that can be transcytosed and systemic infection with small molecules. And I also think that, drug antibody conjugates are a really, interesting opportunity for viruses as well, because you can target it to the virus, even if they're non-neutralizing and they're just hitching a ride into the cell, and then the drug has a mechanism of action inside the cell, interrupting the replication. So, they are, I think, there's a lot of synergy, and of course, people are interested, we and others, in making that happen.
Okay, maybe we have 1 minute left. Last question, just quickly, if you could maybe respond. Everybody's worried about the emergence of these mutants, and I think this discussion focuses on, is that just simply because it's increased transmission, or are the mutants actually escaping the vaccine?
So I think that most of the mutants improve the binding affinity for ACE2. So then they become more pathogenic, more transmissible. You get higher viral loads. There are antibodies that are clearly. There are mutations, particularly outside the receptor binding domain and what's known as the NTD, that are escape mutations. Some are also located in the receptor binding domain. And those, I think, are being generated in long-term or chronically infected patients that give the virus time to replicate and mutate. And then in some cases, they take hold, as you're seeing some of the variants. The good news is they're quite localized, and they seem to come up in vitro selection assays, as well as in infection. So it could be that there's a finite number of ways, again, to toggle that balance of productive ACE2 binding and immune evasion. And I think a lot of the variant vaccines that are being developed by Moderna and others are going to fit well into the boosting of the primary responses that people already have.
Great. Okay, thanks, Case, for the antiviral lecture, and thanks to Andrew for the vaccine lecture.